Alrighty, hello, my name is Abuzar Haider Zaidi, and I'm a Bio 1P92 student at Brock University, and today we're going to be talking about halophilic archaeans, some of the earliest forms of life on Earth and potentially elsewhere in our solar system. So let's start with a brief introduction. First, we're going to be talking about the general domain archaea. It's one of three domains of life on Earth, along with bacteria and eukarya. So when we're talking about subgroups in archaea, we're actually talking about early life on Earth. We'll talk about why that's significant soon, but as for the domain itself, it was established by Carl Woese and George Fox in 1977, before which bacteria and archaea were actually lumped into one domain with the name bacteria, of which archaeans were a group called archaebacteria. Getting a little bit deeper into our classification, we now move to extremophiles. They're able to, as you may have put together, survive in extreme environments, such as hot environments, the autapomorphy for the thermophiles, or acidic environments, as is the case for acidophiles. We're going to be talking about halophiles today, which thrive in salty or saline environments. Breaking down the etymology of the word halophile, we're actually able to derive that they thrive in saline environments. Halo is a Greek prefix meaning salt, and it is combined with another Greek word, phile, which means loving. When we look at the other characteristics of these hypersaline environments in which halophiles thrive, we see a couple of interesting points. They commonly encounter alkaline pH, which sets them apart from acidophiles, but they also encounter higher temperatures, which is actually the autapomorphy for the group thermohalophiles. Halophiles, of course, have adaptations for the environments they encounter. One is the salt-in strategy, in which high concentrations of inorganic salts accumulate within cells, and cells adapt enzymatically to the conditions. You could think of this as the if you can't beat them, join them strategy. Another is the low salt-in compatible solute strategy. A compatible solute is an organic compound of any type. It could be a protein, a lipid, a carbohydrate, but essentially molecules of this compatible solute form a layer that protects the cell from losing hydration to the hypersaline extracellular conditions. You could think of this strategy as the rebellious one. So now we've defined exactly what halophilic archaeans are, but before we dive in, let's establish why they're important to study. I want to focus on the point virtually screaming at you with the bolded red font. So do you remember how I said that when we're talking about archaeans, we're talking about some of the earliest life on Earth? Well, that's part of how we answer the question of how exactly we got here. How is the first life able to survive in the unpredictable environment that was our Earth 3.5 billion years ago? And how is it able to adapt over and over to eventually get us where we are today with an astounding degree of biodiversity on our planet? And how can we act to perpetuate this repeating miracle rather than destroy it? I feel like I've kind of made the other points on here seem really dull, but they're definitely still important. As for the nutrient cycle, we can study how life harnesses energy and the way energy is cycled among life on our planet. Archaea also have industrial potential, notably in pharmaceutical and waste treatment settings. Okay, so let's get to what we've been building up to and introduce the subject of this experiment or this presentation. The isolate that we will be examining today was isolated from a dried up subglacial lake on the red planet Mars. The subglacial lake was hypersaline with high concentration of magnesium salts and perchlorates, which are indicative of the water-induced breakdown of perchloric acid salts. So we know that our isolate is a halophile, but we need to build out a deeper understanding of it to draw meaningful and relevant conclusions. Naturally, this is going to be where we define our purpose. Our purpose here today is going to be to build out a biological profile of the Mars isolate, namely by way of analyzing experimental observations regarding its colony morphology, biochemistry, resistance to antibiotics, and physiology. This is going to allow us to assign an apt name to the Mars isolate, getting one step closer to answering the question of how life on Earth got where it is today, and how it may have been doing the same thing on the planet we're exploring right now. So let's look over how the experimental results were obtained and the context in which they were interpreted. Our control for this experiment was Halobacterium salinarum, a well-researched, Earth-inhabiting halophilic archaean. All results from tests on the Mars isolate were compared to results from the same tests on Halobacterium salinarum. This allows us to put context to the characteristics that the Mars isolate exhibits and again draw meaningful and relevant conclusions from the data. Let's now take a brief look at the methods that were used. For colony morphology tests, colonies of both the Mars isolate and Halobacterium salinarum were grown on solid agar, and the colony's phenotypic characteristics were observed. For biochemistry tests, enzymatic and metabolic end product tests were conducted on both the Mars isolate and Halobacterium salinarum, each with a different method, but they all had defined positive and negative results. For the antibiotic sensitivity test, cultures of the MARS isolate and Halobacterium salinarum were incubated for 2 weeks and then inspected for growth around antibiotic discs. Finally, for the physiology test, colonies of the MARS isolate and Halobacterium salinarum were grown on identical media with one environmental variable, namely sodium chloride concentration, pH, or temperature. So let's begin examining the experimental results. First, we're going to be taking a look at colony morphology of the MARS isolate and Halobacterium salinarum. So this table on the left here looks at the observed colony morphology characteristics of Halobacterium salinarum and the MARS isolate. On the left, we have the features. A green heading indicates that the particular feature was observed to be similar in both specimens, and a red heading indicates that the feature was observed to be different in the MARS isolate. So looking at the column, we can see that there are a pretty even amount of similar and differing traits observed, 4 and 5 respectively, and without getting too detailed, we can also see that the MARS isolate was observed to be relatively similar to Halobacterium salinarum in terms of its colony structure. There was a 20% negative difference in mean colony diameter, but apart from that, it was observed to be similar in form, elevation, and margin. The isolate differed in pigment, which is the most easily observable from the figure on the right, as well as density, motility, and the presence of gas vesicles. One relationship I found especially interesting was that the isolate was non-motile and had denser colonies versus Halobacterium salinarum, which had less dense colonies but was motile. Taken in context with the fact that they differed in pigment, this may indicate that the MARS isolate differs in its means of acquiring and processing nutrients. Let's delve deeper into that with the next section of results, which is biochemistry as well as antibiotic sensitivity. In this section, we're going to be taking a look at the MARS isolate's observed biochemistry and sensitivity to antibiotics with Halobacterium salinarum results for context. These next couple of slides are going to be very text heavy, but the results are relatively straightforward to interpret, keeping the goal of naming the MARS isolate in mind. We're going to be focusing on whether the MARS isolate is similar or different to Halobacterium salinarum. Okay, so let's pick up on our discussion of nutrients with metabolic activity. In Table 2 on the left, we have tests for acid production as a metabolic end product of the digestion of different carbohydrates. Based on the abundance of red titles, we can deduce that the MARS isolate appeared to differ heavily from Halobacterium salinarum and the carbohydrates it was able to produce acid as an end product of. Specifically, the MARS isolate differed in its ability to produce 12 out of 14 of the carbohydrates. Let's continue by analyzing Table 3. Interestingly, while Halobacterium salinarum tested negative for the presence of all the hydrolytic enzymes that were tested for, the MARS isolate tested positive for 4: citrate utilization, esculin hydrolysis, beta-galactosidase activity, and starch hydrolysis. Taken together, it appears as though the MARS isolate's metabolic and digestive machinery differs from that of Halobacterium salinarum. And this hypothesis is established a bit further when we look at the amino acid metabolism of the two samples. While Halobacterium salinarum tested positive for all 5 amino acid metabolism tests, the MARS isolate tested negative for 4 out of 5, the one exception being that it tested positive for urease activity. Finally, regarding biochemistry, the MARS sample tested weakly positive for alkaline phosphatase activity, whereas Halobacterium tested normally positive. While the antibiotic sensitivity data in Table 5 allows us to see practical applications for this research in the case of human infection, we will again be looking at similarities and differences for a purpose of naming the MARS isolate. As can be observed from the text on the left, we can see that the MARS isolate is relatively similar to Halobacterium salinarum in terms of its resistance to different antibiotics. The MARS isolate only differed in its resistance to novabiocin, for which it demonstrated resistance, whereas Halobacterium salinarum did not, and rifampicin, for which it also demonstrated resistance, and Halobacterium salinarum showed intermediate resistance. In our last results section, we're going to be taking a look at physiological results, specifically how the growth of colonies of the MARS isolate and Halobacterium salinarum cultures were impacted by varying environmental conditions, again keeping in mind that we're looking for differences and similarities for the purpose of naming the MARS isolate. First, we're going to be taking a look at the effect of sodium chloride concentration on growth. So for each of the figures in this entire section, we're going to be taking a look at the peak of each graph, which tells us where the optimal growth conditions for the MARS isolate and the earthbound Halobacterium salinarum seem to be. So starting with starting on the left with figure 9, we can see that Halobacterium salinarim's optimal sodium chloride concentration for growth appears to be 4.0 moles, where it produced a mean 600 nm absorbance value of 0.99 with a standard deviation of 0.05. When we analyze figure 10 on the right, we can see that the Mars isolate's optimal sodium chloride concentration for growth appeared to be 3.5 moles, where it produced a mean 600 nm absorbance value of 0.88 with a standard deviation of 0.05. Interestingly, the Halobacterium salinarum culture's 600 nm absorbance value was only 15% less than the Mars isolate at the isolate's optimal concentration of 3.5 moles. For our purposes, we can say that the Mars isolate is relatively similar to Halobacterium salinarum in terms of its optimal sodium chloride concentration. Now, let's talk about the effect of pH on the growth. Starting with figure 11 on the left, we see that Halobacterium salinarum's ideal pH was a neutral 7, where it produced a mean 600 nm absorbance value of 0.92 with a standard deviation of 0.06. This is the same as the Mars isolates, whose colonies exhibited a mean 600 nm absorbance value of 1.0 plus minus 0.04 at the same pH of 7. Going back to our purpose, we can say that the Mars isolate is similar to Halobacterium salinarum in terms of its optimal pH. Finally, let's discuss the effect temperature had on the growth of Halobacterium salinarum and the Mars isolate. We can see from figure 13 on the left that Halobacterium salinarum's ideal temperature for growth likely falls somewhere between 40 and 50 degrees Celsius, probably more towards 50, aligning with our earlier observation that halophiles on Earth also commonly encounter heat in their environments. See how we're starting to bring it back here? Interestingly, the Mars isolate's ideal growth temperature appeared to be considerably lower at 30 degrees Celsius, where it exhibited a mean 600 nm absorbance value of 0.9 with a standard deviation of 0.1. This is a pretty considerable difference, so we can say for our purposes that the Mars isolate appears to differ from Halobacterium salinarum in terms of its optimal temperature. Okay, so let's really bring it all the way back here by summarizing and applying our findings. So what were our key findings? In terms of colony morphology, the Mars isolate appears to be somewhat similar to Halobacterium salinarum at 44% similarity across tests. Regarding biochemistry, the Mars isolate appears to be different from Halobacterium salinarum with only 24% similarity in tested characteristics. The Mars isolate appears to be similar to Halobacterium salinarum in terms of antibiotic sensitivity, with similar reactions across 80% of tested antibiotics. And finally, the Mars isolate appears to be similar to Halobacterium salinarum in terms of physiology, with similar results in 2 out of 3, or 67% of the tests. My proposed name for the Mars isolate is Halobacterium ericium. Since there was a considerable amount of similarities between the Mars isolate and Halobacterium salinarum, I didn't feel it was justified to assign it a new genus. The epithet ericium pays homage to the namesake of Mars, the Greco-Roman god Eris, also known as Mars. He can be identified in paintings by the prominence of the color red in his attire, as is the case with his red cape in this figure here. So that's all for this presentation. Thank you for taking the time to listen. I hope you have a great rest of your day.